Every year, 30,000 Americans die from small cell lung cancer, a devastating tumor type that has become forgotten in the cancer research community. Despite this huge number of deaths, there are few labs that work on small cell, and there's been no improvements in treatments in over 40 years. I'd like to emphasize two features that distinguish small cell from most other cancers. First, small cell is extremely aggressive. By the time a patient finds out they have small cell, typically tumor cells have already spread throughout the body. For these patients with metastatic small cell, they have less than a 1 in 10 chance of surviving for even 2 more years. The second unique feature is how well this tumor type responds to chemotherapy. Tumors melt away in response to chemo. The problem is this is a transient response. Invariably, some tumor cells escape treatment. Those tumor cells expand, and then the tumor comes roaring back in a form that's now resistant to chemotherapy. There's so much hope for improving treatments for small cell because chemo already does most of the work at eliminating small cell from a patient. We just need to find a way to kill the few remaining cells and stop them from expanding to take over the tumor. Mutations in our DNA are at the root of all human cancers. If you are diagnosed with another form of lung cancer, non-small cell lung cancer, your tumor DNA can be sequenced, and if certain gene mutations are identified, you can be treated with a drug that acts to correct the defect caused by the mutation. For small cell lung cancer, we do not have access to such precision medicine or targeted therapy. Because small cell is so metastatic, and because tumors melt away in response to chemo, surgical removal of lung tumors is not a part of small cell treatment. A consequence of this is there is a severe lack of tumor tissue available to researchers to sequence DNA and to identify gene mutations. In fact, for all other cancer types that are major killers, there's been a huge effort to develop a cancer genome atlas of DNA mutations. This is based on sequencing the DNA across many samples and over 30 tumor types. Small cell lung cancer is the 5th most frequent cause of cancer-related death in the US, number 5. But we can't go to the cancer genome atlas for small cell because it doesn't exist. It wasn't included, a consequence of a lack of tumor material. Again, small cell has been a forgotten tumor. I've been running a research program for the last 10 years focused on really understanding genes that drive human cancers. About 5 years ago, I realized how little was being done to improve treatments for small cell, and I pivoted my research program to try to fill that void. I lead a team of 7 researchers, and we are dedicated to finding improved treatments for small cell that will completely eliminate a patient's tumor. Our research finds what genes are mutated, learns whether a mutation is important, learns what that mutation does, and then we find ways to reverse the effects of the mutation using drugs. Our goal is to find a drug that we can add to the chemotherapy to now, in combination, lead to the killing of 100% of the patient's tumor cells so that that tumor can never come roaring back. To identify DNA mutations, we collaborate with lung oncologists at the clinical arm of Fred Hutch, the Seattle Cancer Care Alliance. Because small cell tumors are not surgically removed, we take advantage of the fact that a typical small cell patient will have thousands of lung tumor cells circulating through their bloodstream, a consequence of this tumor type being so aggressive. So when a patient comes to the SCCA for their first chemotherapy treatment, or when that patient comes back to the clinic months later, now with chemo-resistant disease, we get a blood sample. We isolate lung tumor cells from that blood sample and sequence the DNA to identify gene mutations. We now have our gene suspects for further interrogation. We then turn to mouse models to help us figure out which of these mutations are responsible for aggressive and chemo-resistant small cell. We use new genetic approaches where we can introduce into the mouse lung the same mutation that is found in the patient sample. If we introduce an unimportant mutation into the mouse lung, nothing happens. But if we introduce important mutations into the mouse lung, the mice also develop aggressive small cell. By studying how these tumors behave in the mouse model, we can learn why mutation in a certain gene leads to aggressive small cell. By developing a rich understanding of the biology underlying that mutated gene, we could find ways to use drugs to reverse the effects of that mutation. My lab has already identified many mutated genes in human samples, and we have generated a panel of new mouse models for small cell with different combination of mutations that are found in patient samples. Our testing of these mice has led to new potential therapies. Our next goal, and this is where we're especially looking for support, is to establish a mouse clinical trial unit where we can systematically test combinations of therapies for the ability to eliminate small cell in the mouse model. If we find a treatment that can eliminate small cell in the mouse model, we will get together with our clinical collaborators to get that drug into human clinical trials as fast as possible. There are other labs that sequence human tumors to identify gene mutations, and there are other labs that use genetically engineered mice to really understand the biology of these mutations. But for small cell, I don't know of any other lab doing these together. This means that we could rapidly move from identifying a mutation in a patient sample to modeling that mutation in mice and to figuring out how to reverse the effects of that mutation. We're now at the point where we can match the biology of certain mutated genes to drugs that could correct the problem. We're excited to be making real progress towards finding a truly effective treatment with long-term, durable results. Let's end the era of small cell as a forgotten tumor and bring targeted therapies, tumor-eliminating targeted therapies to small cell patients. Thank you.